In this video, we are going to explain the basic principles and general application of flow cytometry. But we have to start with the question, what is flow cytometry?
Well, first we need to explain the goal of cytometry, which is to measure the shape, size, and even specific components of a cell. Flow cytometry measures this by arranging the cells into a shaped flow that let them pass one by one through a laser that measures the size and the scattering of the cells, and also specific components from the cells marked with antibodies. Flow cytometry is sorted with three types of components: fluidics, optics, and electronics. Fluidics components are responsible for aligning and transporting the cells in a single file in order to be analyzed. Optics are a series of detectors and a laser module where the cell size, the scattering of the cells, and the fluorescence of different marked antibodies will be detected. Finally, the electronics is the interface between the sensors and the computer where the signal will be interpreted. Before explaining how the flow cytometer analyzes and presents the cell's information, let's see how the cells are arranged one by one. A turbulent flow is characterized because the particle has a random trajectory, while the shaped flow trajectory is ordered and similar. Thus, a shaped fluid is necessary for the flow cytometer. As the cells flow to the bottom of the container that gets narrower, they start to collect, making the flow turbulent. In order to face this problem, an aerodynamic focusing region is needed on the nozzle design. If the flow has a lot of pressure, the cells will travel faster and will end up colliding. So in order to avoid a turbulent flow, the pressure control is also important. In order to deliver a single cell to the optics components, a sample with a stain cell is needed. A sheet fluid of physiological buffer will flow independently from the sample. Then the tubing, pumps, and valves will deliver the sample suspension into a single file stream of cells in the nozzle, with the sheet fluid straight to the optics.
Now we are going to talk about the optics that follow flow cytometry. In flow cytometry, we can measure fluorescence, side, and forward scatter. Forward scatter detector is in line with the laser. On the other hand, fluorescence and side scatter detectors are at 90 degrees from the laser. When light passes through the sample, it's going to scatter into different directions. Forward scatter measures the diffraction of light around the cell, so it is related to its size. If cells are bigger, the signal obtained is going to be higher. Side scatter tells us about the complexity of the cell. The detector measures the light that is reflected by the cells. The components that increase side scatter are the nucleus and granulocytes. In this example, neutrophils are going to have a higher side scatter signal because of the form of their nucleus and granulocytes present in the cytoplasm. We also use antibodies or fluorescent dyes to detect molecules in the cell. Fluorophores are excited by their specific wavelength when they pass through the beam. The light emitted is directed through a pathway where it is going to be filtered and is going to reach the correct detector for their measurement. This was illustrative to understand the principle, but in practice, things are a little bit different. Fluorophores should be selected very carefully, like in this example. This spectrum shows fluorophores that can be excited with a laser of 488 nanometers, but each one is going to emit in different wavelengths. Now, how do the cytometer detect them? The detectors have a specific configuration on the cytometer, like in this arrange called octagon. In each circle, there is a detector that has two filters. When light enters to this arrange, it will encounter a filter. The first one is a dichroic low-pass filter. This means that wavelengths below 670 nanometers are going to be reflected, and wavelengths above this value are going to pass through. The second is a bandpass filter. The numbers mean that centering in a wavelength of 695 nanometers, we are going to have a bandwidth of 40 nanometers that are going to reach the detector. This is going to happen in each detector. Finally, we are going to have this filter that is going to detect side scatter signal.
Here we have a case where we want to identify neutrophils through the expression of CD16B and CD15. The first step consists on the staining of isolated granulocytes using fluorescently labeled antibodies. Then the sample has to be placed in the sample inlet of the flow cytometer, and now the equipment will start to update the sample. Next to the flow cytometer, there is a computer that is receiving the electronic signals that we can visualize by using a software such as FACSDiva. The data can be plotted using histograms, contours, and dot plots. The deep data analysis has to be performed using other software such as FlowJo. Here we have purified granulocytic population. We have to plot the unstained population using forward and side scatter parameters. This will allow us to separate the population by its complexity, and once we have this population, we have to take only the single cells. We do this by using forward scatter area versus height. Now we have to set our negative population to our fluorochromes, FITC, and Brian Violet 21. Then we copy all the gains of the unstained population and we have to apply it to our stained population. We adjust the gains to round all the population, the granulocytes, then the singlets, and here are neutrophils, our positive population to CD16B and CD15. Now we know this, think about the limitless applications flow cytometry can be useful. We can characterize beads, bacteria, and all kinds of eukaryotic cells you can imagine. We hope this information helped you if someday you have to work and analyze flow cytometry data. See you!